Our discussion of myasthenia must first start with recalling how the nerves and skeletal muscle interface in healthy patients. The first component of the neuromuscular junction is the neuron. Neurons that innervate skeletal muscle contain vesicles full of the neurotransmitter acetylcholine, shown here in green. As an impulse travels down the neuron, it triggers an influx of sodium. This sodium signals acetylcholine vesicles to prepare to bind to the cell membrane. Meanwhile, across the synaptic cleft lies a muscle cell. It has lots of deep grooves in its membrane that increase its surface area, which is peppered with acetylcholine receptors. Each receptor is actually a sodium channel made up of 5 subunits. While acetylcholine is not bound, it remains in the closed position. At the neuron, sodium influx has triggered acetylcholine vesicles to bind and merge with the cell membrane. This allows vesicles to dump their acetylcholine into the synaptic cleft. Acetylcholine then diffuses across the cleft and binds to acetylcholine receptors. This opens the receptor channels and allows sodium to rush into the muscle cell. In summary, this triggers muscle movement. The enzyme acetylcholinesterase, shown here in purple, will then remove and degrade acetylcholine from the muscle receptors. But in most types of myasthenia gravis, IgG antibodies attach to the acetylcholine receptors and block acetylcholine binding sites. This begins another sequence of events. The muscle-specific tyrosine kinase, or MuSK protein, is an intramembrane enzyme that is typically responsible for destroying old or faulty acetylcholine-gated sodium channels in healthy cells. In myasthenia gravis, the MuSK protein recognizes malfunctioning IgG-bound acetylcholine receptors and signals the muscle membrane to internalize them into vesicles. They are subsequently destroyed so that they can be replaced by functioning receptors. In addition to blocking acetylcholine receptors, the IgG in this type of myasthenia also fixes complement, a series of proteins that, when activated, increase membrane permeability and can lead to cell death. This myasthenia, shown in red, accounts for about 85% of all myasthenia gravis cases. It is often correlated with thymic abnormalities like thymoma or thymic hyperplasia. But there's another type of myasthenia we have yet to discuss. This type is caused by antibodies that target the MuSK protein or other unidentified postsynaptic targets, and accounts for the remaining 15% of cases. They do not activate complement, are not usually associated with thymic abnormalities, and this myasthenia is typified by more focal signs and symptoms of disease. This antibody binding inhibits the action of the MuSK protein and prevents proper turnover and replacement of the old acetylcholine receptors. Ultimately, the results of both these types of myasthenia result in the same outcomes. The synaptic cleft gap is widened. Receptor density decreases as blocked receptors are destroyed faster than they can be replaced, and constant internalization of receptors leads to decreased membrane surface area as the muscle loses its grooved appearance.